Hi, I'm Leona.
Hi, I am Sam.
We both work and study here at the Department of Computational Biology at UNIL.
And we are interested in the biology of aging.
When you think about aging, many things come to your mind. From getting wrinkles, grey hair, wearing glasses, and maybe even death.
Aging and life expectancy are complex to study. You can estimate your biological age by looking, for instance, at your lifestyle, at your health, but also by looking inside your cells and measure the length of your telomeres.
Telomere length? What's that?
Our bodies are made of cells that contain a nucleus in which we find our DNA. Telomeres are repeated non-coding DNA sequences located at the end of our DNA strands. The fact that they are non-coding means that they do not contain any information.
If they don't contain any information, why do we have them? And how do they impact our aging?
Over the course of our life, most cells divide many times. Each time a cell divides, the DNA gets a little bit shorter. The telomeres act as protective caps so that the coding parts remain unaffected. At the same time, the telomeres work as an internal clock. When a cell's telomeres grow too short, the cell goes into apoptosis. So it basically commits cellular suicide.
Oh wow, so if we could extend the telomeres, would we be immortal?
Well, it's not that simple. Cells undergo apoptosis for a reason. If they grow too old, they might accumulate many genetic mutations, which might lead to an interruption of their cellular functioning, but can also lead to cancer.
Okay, so longer telomeres are not necessarily a good thing, but can also bring risks. Would you still say that shorter telomeres drive aging, or is it maybe just that shorter telomeres result from aging?
You know what? Let's go to my office to answer this question. Okay. While too short but also too long telomeres have been associated with diseases, on average, people with longer telomeres seem to live a bit longer. This association has been found in multiple studies, and we also looked into it by using a method called Mendelian randomization. By applying this method, we also found that longer telomeres may lead to longer lifespan. But if you want to know more about this method, check out Lisa's video here.
This is so exciting! If we know that longer telomeres lead to longer life expectancy, do we already know how we can keep our telomeres longer over time?
It would be nice to really know, right? But it's complicated. Let's look at smoking, for instance. Online, we find that smoking is associated with shorter telomeres. But we also find some Mendelian randomization studies suggesting that this relationship is not causal.
Okay, but how can there be an association if it is not causal?
Let me visualize that. We have smoking, which is associated with shorter telomere length, and using Mendelian randomization, we try to see if smoking directly causes telomeres to shorten, because this association could also be what we call spurious. For example, if there is some other factor that both leads to smoking and telomere shortening, and we do not account for this, it looks as if smoking was directly associated with telomere shortening. So the strength of the Mendelian randomization analysis is that it to some extent controls for such factors.
Okay, so if MR, the method that you've mentioned before, does not find a causal link between smoking and telomere length, that might indicate that there are confounders, like other factors which have an impact on both smoking and telomere length. Did you already look what potential confounders might be?
I'm on it right now, but this would clearly exceed the scope of this video.
Okay, so in brief, telomeres are the non-coding ending of our DNA. These get shorter with each cell division and thus over the course of our life. Longer telomeres tend to be associated with a longer life expectancy. To precisely understand which factors accelerate telomere shortening, we need more research and another video from you. So, with increasing life expectancy, telomere shortening may play a crucial role in healthy aging. By understanding which factors causally contribute to telomere shortening, we might be able to promote healthy aging.